welcome to Coffee Chats with Experts. My name is Laura Moriarty and I'm joining you today from California. This coffee chat this time around is around Bio-Plex multiplex immunoassays and their use in vaccine research and development. And before we kick off, I'm going to introduce you to our lovely panelists today. So starting off, we have Candice Cox. Candice is also joining us from California. So Candice started at the Bio-Rad team as part of assay development in R&D. So she has had her hands in the assays, so to speak, and has lots of experience. She now works over at leading the marketing group for the Multiplex Immunoassay business. Welcome, Candice.
Thanks, Laura.
And also joining us today, we have Rick Ando joining us from the East Coast, I believe, Rick, right?
Cambridge, Massachusetts.
Lovely. Nice to have you. And we're actually very excited to have Rick join us. He has been in the field supporting Bio-Rad customers and scientists like yourselves for over 15 years. So he brings a lot of experience and he's a senior proteomics field application scientist. So we're really excited to have you join today. Thanks, Rick.
Thanks, Laura.
All right. And then finally, filling out the panel this morning, we have Brett Hauser. Welcome, Brett.
Hi.
And so Brett is also joining us. He's based in California. He is a senior global product manager for the immunoassay business. And he has been working supporting customers doing Bio-Plex assays for over 8 years. And specifically, he has been involved in a lot of development for the software, which we all love, right? And reagents as well. So welcome, Brett.
Thank you.
All right, so before we start, I want to give a couple of housekeeping thoughts to everybody who's listening in. So we love to get your questions. That is why we're here this morning. We want to hear from you. So please do type your questions into the Q&A and we will address those to the panel and hopefully get you a great answer and some ideas to help troubleshoot any concerns you may be having in the lab. Now, if you're listening to this on demand, then don't worry, you can still get your questions answered. Just type them into the chat as well, and we will get those over to our experts and get you an answer that way. So either live or on demand, we are here to address your concerns. So with that, I would like to kick us off. And as always, I give a couple of questions from myself, just to kind of get the panelists woken up and get their brains going a little bit. So hopefully this one's not too hard for you. And honestly, if you can't answer this first question, then you shouldn't be in the panel. All right, so here we go. All right, so what are multiplex immunoassays and why are they used in vaccine development? All right, so with this very hard question, who would like to kick us off? Okay, Candice.
I'll go ahead and do that. Yeah, so for those of you guys who are very familiar with ELISA formats, multiplex immunoassays are very comparable and have a lot of the same configurations. So since most of our Bio-Plex assays are sandwich assays, I'll go with that format to explain it. Everything's happening in a 96-well plate, but instead of everything being absorbed to the bottom of the well, everything that you're capturing is on a bead. And so you have your capture antibody coated onto that bead and everything is in suspension. And so the rest of the format of the assay is very similar. You're capturing your antigen with another detection antibody and you're basically able to see it through some sort of reporter molecule. In our case, it's phycoerythrin. Oftentimes for ELISAs, it's colorimetric. So instead of OD, you're looking at median fluorescent intensity. And then the way that you're able to combine multiple targets and analyze them in the same well is because of the bead. And so the core of the technology is really the bead, where you have two different ratios of dyes, where that is a specific spectral address that allows you to basically designate that a bead is, for TNF-alpha or IL-6. And so that's really designated by whoever's developing the assay or by the vendor. And so they're very useful for vaccine development because of the flexible formats. So one way that they can be used is basically by coding the antibodies onto the bead, looking at cytokines. And so, as many of you guys know, cytokines are prolific in terms of the immune response and can very much give us a big picture of what's happening. So immune response profiling is one big thing. And then another way that they can be used is by coding the antigens that are very specific to that microbial thing, or I guess the pathogen that you're trying to vaccinate against, so that you can look at the antibody response of the host that you've actually immunized. And so by doing that, you're able to look at basically antibody titers, and then also look at that they're very specific to the immune response that you're trying to elicit with your vaccine.
Awesome. Wow, I think, yeah, that says it all. So Brett or Rick, did Candice miss anything there? Do you guys want to jump in?
Oh, I just wanted to add that, when the question comes about multiplexing, in this case, you can multiplex up to 100 different bead regions for the Bio-Plex 200 and then 500 bead regions for the 3D. So that's kind of the plex level capabilities.
Mm-hmm. Awesome. So for folks out there who are doing, a bunch of ELISAs, maybe this is an option, right? Yeah.
Exactly.
That's certainly when I was in the lab, that's certainly the reason I did it, was to be able to see lots and lots of things at once. All right.
One thing that I would add before moving on to the next is that the Bio-Plex platform, one of the nice hallmarks of this multiplex assay are really the arenas of targets that we have. Many are off the shelf, or many are pre-designed assays for groups of biologically relevant targets. But then there's the whole field with ELISAs that you develop on your own. You can make custom assays. So the platform is very adaptable as any immunoassay, as Candice mentioned.
Nice. Fabulous. All right, so I think everyone has stretched their brain muscles and is ready for something a little bit harder. So I think this one, I like this one as well, because it's very topical. So in your thoughts, what do you think around the use of multiplex immunoassays to look at patients who've been exposed to coronavirus?
Well, I'll jump in on this one. And now is a great time to talk about this, because so little is really known about coronavirus and COVID-19 virus infection. So, a lot of the discovery and research is surrounding what kind of immunogenicity and the immunoprofiling of a infection. So this kind of multiplex platform lends itself very nicely. Even, just learning the basics of a cytokine reaction to the infection. And maybe as we understand more, by using some larger targets, we can narrow it down, focus in on specific antigens or specific key players in the response, and even break down different populations as maybe a predictive measure or different higher or less risk categories of different patients. So, for instance, I mentioned a large panel. We have a 48-plex cytokine panel, and there's already been a lot of work done on establishing some of the early on responses upon infection. So,
that's great.
Laura?
Go ahead, Candice.
Oh, okay. I was just going to say that, and with the speed of the discoveries being made today, I kind of feel that the multiplex immunoassay format really helps with looking at a big, panel and looking at many different proteins in a much faster way than you would if you were looking at them individually. So it really compresses the amount of time that you're spending, measuring all these different analytes. And so with as fast as everything is going today with the whole COVID-19 vaccine development, I think that it really is a good tool to have in, your toolkit.
Yeah, and speaking of fast, there was really an interesting publication that came out in January in the Lancet, where scientists were looking at cytokine response using the 48-plex in patients, starting in November. So you can imagine, symptoms coming in, and they were looking to see how the cytokine level profiles may predict if a patient needs to be in ICU or not. So it was kind of interesting, but also interesting in the fact that it was able to be published so quickly.
Yeah, there's a lot of great research going on right now. I'm reading those pre-proof or preprint publications. There's a lot, which is great. Collaboration is key for science, moving science forward. And it's a crazy time, but yeah, pretty hopeful with everything that's going on. All right, I'll get off my soapbox and get us to another question. Ooh, this is a good one. Okay, here we go. All right, so what sort of adjuvants are being used in COVID vaccines? Are people making new ones? Can multiplex immunoassays be used to test? Can be used to test which adjuvant is best. This is nice. Anybody want to take this? Candace?
I can start that one.
All right.
So, before moving in, just to, set the stage, adjuvants are things, basically the formulation that you're going to put your vaccine in that's going to inspire higher levels of immunogenicity so that you can basically develop a stable immunity or stable immune response by developing, memory B cells and all of that. So you really want to provide a dose with the adjuvant that's going to allow for you to recruit, the players that's going to elicit higher immunogenicity with low toxicity. So in terms of COVID-19, what I've seen out there is GSK is actually providing over 1 billion doses of their adjuvant that they've used in a past vaccine. So about 10 years ago, H1N1 swine flu, epidemic. And so in that case, they had developed a vaccine, AS03, and they're actually using it in conjunction with some of the vaccines that are being developed out there. And that's really going to help with the speed of development of vaccines because they've already established a safety profile for that specific adjuvant. And so I think that that's a really nice thing to see that people are collaborating on this. I read also another example where Dynavax is providing their adjuvant, which was used for a hepatitis B vaccine that they had already developed to another company. I think it's Sinovac. And so that's really going to speed up that establishing the safety profile for vaccines and really going to push them through the clinical trials a lot faster. So that's really nice to see. And then in terms of just studies that I've read about where multiplex immunoassays, specifically Bio-Plex, have been used to look at adjuvants and the effects and immunogenicity. Gardasil is an example of basically a vaccine for HPV that has been used. And early on in the development, they looked at basically in preclinical phases, different formulations of their aluminum adjuvant and the response in rhesus macaques. So that was a paper that was published a while back, but there's definitely established work that's been done using this platform, looking at the different formulations.
And is that aluminum or aluminium, Candace?
Oh, I think it's aluminum for California and the US.
Sorry, I just had to say. Anybody have anything to add on that one? Nope. Oh, nice. All right. So let's move on from aluminium discussions to sorry. It's a scientific joke, right? So it's okay to say it. All right, so here we go. Regarding antibody specificity and ability to bind a specific target or its fragment. Okay, think about that. What is the difference between an ELISA and a multiplex assay? Ooh.
I could handle that, and just by saying, really, at some levels, there may not be too big of a difference. The same principles that you use to create an ELISA are used to formulate a Bio-Plex assay. There's a lot of advantages, obviously. You may, in fact, use the same antibodies to coat the beads as you might to coat the well of a 96-well plate. The differences are going to be in the reporter, as Candace mentioned, it's a fluorescent assay versus a colorimetric assay. The machine that we're reading it on, obviously, is very different, much more like a flow cytometer as opposed to a spectrophotometer. But it's the ability to coat these antibodies onto multiple beads within the context of the same assay to allow you to get a multiplex panel going and analyze a lot of antigens simultaneously.
I was going to say, what about cross-reactivity?
Oh, well, yeah. So cross-reactivity is, are you asking me?
I was asking you, but
something
I can answer too. I was just.
Yeah.
Yeah, why don't you pose and answer the question?
Yeah, go on, Brett.
So you might wonder, what about cross-reactivity? So yeah, that's another, that would be a little bit of a difference between ELISA and multiplexing, is that you need to consider, your antibodies to be very specific and that they don't cross-react. And to do that, we typically just do cross-reactivity studies to validate and verify. And of course, with ELISA, it's single plex, so you don't do that.
Yeah, and just to put it in context, sorry, I didn't raise my hand. And just put it into context, I think Brett mentioned we have a 48-plex, so that means that there are 48 different antibody pairs that are playing together in that one well, and we've tested the cross-reactivity between all of those different assays. So we know that they're highly specific, and for multiplex immunoassays, that's one of the criteria that is a must in order to get quality results. And so in a way, I would say that we have a lot more stringency around specificity when it comes to multiplex immunoassays, because we actually are testing for it and provide that information for all of our different assays within our panels.
All righty. Okay, let's find another question for you. Here we go. Oh, this is another vaccine one. Okay. So what ways are vaccines evaluated to ensure they are safe and non-toxic? Who wants to take this one? Candice?
Take that. Sure. So like any other therapeutic that's developed out there, they go through the same process in terms of preclinical all the way through different levels of clinical trial, 1 through 3. And so for preclinical, you're really trying to establish whether or not your formulation, your adjuvants, everything that the vaccine is going to be injected with is basically non-toxic at that point. And you're trying to establish the dose at which is going to prove to be efficacious. So when you're characterizing vaccines, you're looking at other things like potency and purity, but where multiplex immunoassays really come in are looking at the safety and efficacy. And so in that preclinical phase, when you're looking at different models, you could be looking at it in, a specific type of cell line. So, basically analyzing cell culture supernatants to understand whether or not you're getting a negative response. And then you're trying to find the right animal model as well to make sure that the profiles that you're looking at of cytokines or any other effects. So the FDA really outlines this very well in terms of the types of testing. So there's, toxicity around reproducibility, reproduction, mutagenicity, carcinogenicity, and also safety pharmacology. So those are the things that you're doing in that preclinical phase. But once you establish a good dosing regimen and you sort of have a range of the time points that you want to administer that vaccine, then you're going to go into humans. And so for that first phase 1, it's pretty small in terms of the number of participants or volunteers that you have. But at that point, you're really looking at dosing and the different levels of dosing that is required to get the efficacy that you want for your vaccine. So I actually recently read, in terms of the Moderna vaccine, that one of the patients that had an adverse event reported, basically it was just, they ended up getting, dizzy and having a fever or something like that. But they actually ended up being in that group that got the largest dose. So it's not necessarily anything severe. He got over it after a few days, but they want to note that kind of thing, in terms of what can be a really bad outcome in that first phase. And then as you go through phase 2 and 3, the population of your trials gets bigger and bigger. So you're able to see how the immune response is in a larger group of people with regards to how efficacious your vaccine is. And you're always all throughout looking at safety. And that happens even with post-market surveillance. A lot of research institutes and a lot of groups are out there that take note of adverse events, as well as study the after effects of vaccines well beyond their time of launching into the market.
Brilliant.
Yeah, I'd add that the whole purpose of a vaccine is to give somebody a large immune response. So in doing that, you want to moderate that response to get the desired effect without harming the patient. In a lot of cases, a vaccine is going to be delivered to healthy people. So you have to do a lot of safety testing behind the scenes to make sure that the general population is not going to be worse off following a vaccination beforehand.
Yep.
Right. Yeah, immunogenicity is a good thing when it comes to vaccines.
Yeah, it's funny. It's like opposite to what we're used to.
Yeah, talking about therapeutic.
Right. So here's another one. Will we need to optimize the assays for running patient samples infected with COVID? Okay, so I'm guessing this is Bio-Plex Immunoassays. What are your recommendations?
Okay. So I just spoke to some people who were doing research around that. And one of the things that they had mentioned are heat inactivation, definitely working in a biosafety cabinet during sample preparation. And so sometimes if, say, like you're looking at samples, not necessarily from like a hospital or something, but you're looking at it for doing just some basic research. Sometimes the vendors will also do the inactivation for you so that when they send it to you, you can actually just have a virus positive sample, but that is more safe to use. And then Rick, I don't know if you had, were you raising your hand as well?
Oh, well, I would say, you know, the normal laboratory precautions, I would say, with regard to COVID-19. I've been in many labs where they're studying HIV and other very communicable diseases as well. And standard laboratory precautions when working with these assays is pretty commonplace. I would say there's nothing too out of the ordinary one would have to do studying this disease versus many other patient serum samples.
Yeah, and then just to mention as well, go ahead. Sorry, Brett.
I was just going to add that as far as the instrument goes, you know, it protocols a 10% bleach solution.
For this, yeah, for the instrument.
Yeah. And I thought I read in the tech support files, but no, go ahead.
No, go ahead. Yeah.
Oh, was there a recommendation for 2% para formaldehyde solution?
That's right.
I thought I read that in a tech support case to neutralize posts just prior to reading. Oh,
I was talking more about fixing the assay after it's done, prior to putting it, you know, prior to taking it out. Yeah.
Mm-hmm. Yeah, the main contact when dealing with samples, obviously when you're opening the freezer, maybe making aliquots, maybe pipetting them or diluting them in anticipation of adding them to the plate. But then once they're on the plate, you do a series of washes, either manually or using a automated washer. You can treat the waste coming off that wash machine as contaminated with 10% bleach. And then at the very tail end of the whole assay, upon reading the plate in the instrument, there is a waste line that dumps into a bottle there that can be treated similarly.
Brilliant. All right. So here we go. Here's a good one. So this person's asking, they use the Bio-Plex assays in their preclinical lab, preclinical assays. Can they use them for clinical trials too?
So I'll go ahead and start the answer to this one. The short answer is yes, because the system is completely flexible, so you can certainly scale up very easily in lots of different ways. It's amenable to automation so that you can process lots of samples through them. So when you advance in your clinical trials, your sample sizes become in the thousands. We have certainly quite a few publications where people have processed 5,000 samples using the Bio-Plex system or the Linux platform. So that's all I had to say.
And I'll just add specifically menu and analytes availability. We do have rat and mouse cytokines that are very comparable to our offering for human cytokines. So when you're doing that immune response profiling, then you have very similar targets to assess between your animal model of choice and also your patients that you choose to evaluate during the clinical trials.
Yeah, that's important. You don't want to have to go back and revalidate a bunch of different markers. So awesome. But making it easy, right? As easy as possible, right?
As streamlined as we can.
There you go. All right, here's another one. Okay. So what types of positive and negative controls, this is really important, are essential to obtain when developing an in-house MIA, microsphere-based immunoassay?
Well, I can jump in. When designing your own assay, essentially, you are the one who's going to be providing antibodies, right? So capture antibodies and also a detection antibody. And you need some kind of antigen between there, which is maybe going to be provided by the sample or for a standard curve. So when you've got your assay designed and cross-reactivity tested and so forth, when you're actually running it, the controls that you're going to have may either take the form of purified protein, purified antigen, or they could be patient samples with known high or low levels of the suspected antigen. So it can be a variety of different ways, but just something that can be run periodically to either verify the assay or comparable results from day to day using the same assay on different patients.
That's right. And just to add one last comment to that, just in terms of the matrix and what type of biological fluid you're using, you want everything to be as comparable as possible. And then in terms of the ranges in which you expect to see the levels of that specific analyte that you're looking at, you might want to have controls close to that level, or a biological sample like Rick had mentioned is the best, whereas sometimes when they're at very low levels, you can use that same matrix that the sample is very similar to the sample, but just spike it. So that's another option for controls in terms of positive.
Yeah. Oh, this is a good one. So are multiplex immunoassays used in QC'ing manufactured products? What is the benefit over an ELISA? Who would like to take this one?
I'll start. Sure, yeah, I'll go. We have customers that do use them in manufacturing. One of them is one of the largest manufacturers of vaccine in the world. The way they use them is several different applications. One is immunogenicity, where they're looking at total IgG to look at the potency of the assay. They also look at, they create their own competitive innovation assays to characterize the antigens that they're using. They take a look at the specificity of monoclonal antibodies using the system, and I believe purity as well. So they're definitely being used in manufacturing of the vaccines themselves, in development and manufacturing. Anybody have anything else to add?
Yeah, just in terms of the benefits over ELISA, I think that, the assay, the amount of information that you're getting, it could be a savings of like 3 weeks when you're looking at a large screening panel in terms of how long that would take you to run everything on ELISA day after day. And then in terms of animal models that we were talking about, maybe for the preclinical phase, that's when the sample volume is so critical. So you might have to make some compromises in regards to what are the analytes that you'll be looking at, whereas if you are using a multiplex immunoassay, you're able to look at many different analytes. So for our rat panel, it's like a 23 different analytes within that well in a very small volume that's usually less than 15 microliters. So just the sample savings itself. And then in terms of analysis, I think that having everything on the same plate, run with the same buffers, same day, really reduce those confounding variables that can contribute to differences that may not be due to biology. So I think those are, some of the benefits that you have looking at things all in the same well with Bio-Plex assays.
Yeah. Some great points there. Thank you. All right. Here's a good one. Can someone use the plate more than once? For example, divide the plate twice. I guess divide and use the plate twice. What's your recommendations on that?
Yeah.
Go, Rick.
I'll take that. Just, at a level when you're benchtop. Typically, it's a one shot per plate kind of assay. You don't want to actually reuse the beads, but the beads are aliquoted upon final dilution to a defined concentration. Giving an example, dividing the plate twice or something might be related to running over-the-counter assays, perhaps in a 384 well format. I've heard some laboratories splitting the assay and running a half a plate of a 384 versus 96 or something like that. Sometimes you always get the question regarding reuse of a plate when you're only using maybe a portion of the plate for some samples and then want to go ahead and use the other portion of the plate later on another day or another set of samples. And in that case, I would basically just buy a box of plates instead of trying to preserve one half for later use. The plates that we use are a pretty standardized plate, which is very thin-walled on the bottom to allow as strong of a magnetic attraction as possible, and they're pretty common. So the plate is not the limiting factor. It's obviously the reagents, the assay, and the components to it. And most, if you're purchasing them, come in the format of a 96-well assay.
And I'll just add that we've seen some really weird things happen with standards. So we really don't recommend reconstituting the standard, putting it in the fridge and using it over self-replace. It needs to be used fresh. If you do that, then you'll get some very strange results, and it's really not worth it.
Yeah, and so with regards to that, we do sell standalone vials of the standard. So these are lyophilized vials of standard, so that if you are planning on doing this, best practice would be to purchase another vial of standard to go ahead and run that second half of the plate. And then the other thing I just wanted to mention, because I think, Rick, you said something about, the magnetic aspect. So the beads are actually magnetic beads. So all of the separation that you're doing with the washes after each and every step in the assay allow you to do that in an automated fashion or using a handheld magnetic plate washer. So just to give everyone some context to that.
Mm-hmm. And just a very simple answer to this question would be to plan your experiments to basically use the full plate in one shot. It's best to go and use everything all at once and plan accordingly. Typically, samples are run in duplex to kind of conserve versus threeplex your real estate on the plate and also get a little bit of replicate statistics. And generally, you can get about 78 on average samples per plate with controls, with a blank, with a standard curve, and so forth. So 78 divided by 2 for duplicates.
Can you divide 78 by 2, Rick?
39.
Nice. All right, just checking. All right, here we go.
It's later in Boston. He's been away for hours now.
That's true, yeah.
Don't ask me that question right now.
I know, because I couldn't answer it. Also, what type of samples do you recommend to run Bio-Plex with? Do you think serum would be a good choice, knowing that most of the assays start with nasal swabs and co? I'm not sure what the co is, but yeah.
I can jump in. Just most of the assays are certainly validated on biological samples, serum, plasma. If you're going in vitro, generally cell culture supernatants and so forth. But with regard to the question, nasal swabs, I'm not sure if that would be a great type of sample. Of course, you can solubilize a swab and get some sample off of that. But I believe that those type of nasal swab tests are often used for the real-time PCR, looking for DNA or RNA of the virus, as opposed to antigens found perhaps as a reaction to the infection and so forth. So depending on what aspect of the disease you're trying to look for, that will obviously dictate what type of sample you're looking for or looking at.
Yeah, good point. Good points. All right. So here we go. Here's another one. Boom. Coming to you. This is a long one. Okay. One of the challenges in MIA for serology purpose is finding that sample high enough for all of the antibodies of interest. Ooh, what is the highest multiplex assay for antibody screening that you have ever developed? Ooh, and how do you look for a reference standard curve sample? How do you test cross-reactivity? So this is a big question. So who wants to kick us off first with this one? Maybe we can start with the biggest one we've ever developed. Yeah.
Well, I was going to jump in and just talk, the fundamentals of the technology are such that, depending on the instrument that you're reading, it's either a 25, a 100, or a 500 plex assay. So this is the upper limit of the beads that are all uniformly coupled to comprise a Bio-Plex or Luminex type of test. In practice, you're never going to have a 500 plex protein type of assay, just the opportunity for cross-reactivity, all the antibodies, 500 capture, 500 detection, 500 antigens. It just simply is not going to work. So assays do fall apart in terms of sensitivity or cross-reactivity at those very high plex levels. It's relatively routine, and our biggest panel for many years, for example, was a 27-plex human cytokine panel. We've recently expanded that, our largest commercially available one, and this is a good rough estimate of the upper limits. Around a 48-plex, 48 to 50 is probably the upper limits that one would want to design one for. You could probably go higher with a little more stringent cross-reactivity testing. But in reality, those large panels are very useful in the discovery stage of a lot of processes. A lot of times you're hopefully whittling it down to a lower subset or a smaller number of antigens or tests of interest to be included in a final type of assay. So.
Can I add something to that as well?
Yeah.
So the question was about the serology tests. So in the life science Bio-Plex product portfolio, we don't have a lot of serology tests. We do have an isotyping test where we measure different levels of isotypes where you're actually capturing an antibody. But I think that the question here has to do with the specific targets being coded on the bead and then looking at the levels in different patient samples. So the Bio-Plex 2200, which is on our CDG side, have a lot of assays that are in this format. And if I recall, I think they have one that's maybe up to a 10-plex in terms of one that they've established. Don't quote me on that, but I think that they have one around that level. It just depends on, I guess, how specific, not specific, but how available antibodies would be that you could basically use for a standard curve if you wanted to make it quantitative. Usually, I think they just end up having non-quantitative assays, and they just have levels and thresholds. But if you wanted to create a reference standard curve, you would have to basically have an antibody that you knew recognized that specific antigen that you had coded on the bead. And then you could go ahead and try and titrate that down to get a range where you think your levels physiologically would be for that antibody. And then in terms of cross-reactivity, you would basically have all of the different beads that have all of the different antigens coded onto there. And then you would use that specific reference antibody against each of those bead sets that are all mixed together to make sure that you're only getting signal for your specific assay that you are looking at. And so you would do that individually to make sure that the levels correspond to when they're all together as they do when they are, you're looking at each of them individually. So that's how you would test the cross-reactivity if you were trying to develop a serology assay. So we could talk about theoretically, but the best, I guess, probably reference of something like that today in this format using the same technology is are the assays on the Bio-Plex 2200.
And for those of you who aren't familiar with all the Bio-Rad acronyms, CDG is the clinical diagnostic side of Bio-Rad, and the folks on the call today are from the life sciences side. And the Bio-Plex technology, we have instruments and assays that are both for use in life science and on clinical diagnostics. So, all right. Okay, enough of my spiel.
Has the acronym handbook?
No. They do give you one when you start at Bio-Rad, so it is helpful. All right, here we go. So do various antibody affinities attached to the beads affect the overall outcome? Who wants to take this one?
Well,
Go, Rick.
That's really fundamental to the whole assay. So the affinities that each of the antibodies have are really going to detect or determine your lower limits of quantitation, your dynamic range of the assay, and really your effective working range or your sensitivity. So if you're fortunate enough to have a handful of good antibodies towards a given antigen, whether they're monoclonal or even a couple polyclonals, it's good to test them in combinations where you're coupling some to the beads, using others as your biotinylated secondary, and mixing them up to determine which combination gives you the best dynamic range and sensitivity. So in the design of these assays, that's definitely something to look into and maybe optimize if you have the opportunity to choose among possible candidates.
All right, let's move us along. It's another long one. Okay, so can you describe the assay to test for various antibody isotypes after COVID disease? The antigen or spike protein coated to the bead, add patient sample, add the beads coated with the antibody to detect various isotypes, and is this a kit? So basically, how would you design the assay? Go ahead, Candace.
Yeah, so the question actually describes a lot of the steps already in terms of what you would be coding onto the bead. So yeah, spike proteins can definitely be coded on there. Another, I think, antigen that I've seen out in ELISAs that are out in the market are the nucleocapsid. So you basically coding it with things that elicit an antibody response in different patients. And so you could start there and you would code each of these different antigens on a different bead region in order to be able to decide, or detect which one your antibodies in circulation are recognizing. And then your detection would be the anti-isotype. And so your detection would be either anti-IgG, anti-IgM, anti-IgA. And so at this point, even in the multiplexing format, you would have to have those be separate because there's only one reporter laser on the 200. And so you would only be able to detect, distinguish between what's coded on the bead, but not necessarily have 3 different anti-isotype antibodies. So you would have basically a 3-plex if it was the 2 spike proteins in the nucleocapsid with IgG, and then one with IgA and IgM specific kits. So right now, there's not anything on the market. There are ELISAs that look at different isotypes, but they're not able to have different antigens in that well necessarily. They're looking at one antigen at a time.
All right, thank you, Candace. Okay,
You're welcome.
A couple more here. So this is a good one, I think. This is, this is good advice, I think, for anyone who's setting up an experiment, really. How do I compare sample data collected across different days, and what controls can I use?
I can kick that off too, just for my days in the lab. Why not? So you could really look at the standard curve. So if you're going to do or develop a quantitative assay, you'll be developing a standard curve. So we could take an off-the-shelf assay that we have. So for cytokine assays, we have a standard curve for each and every cytokine that's in that panel. And so we have a range in which we can basically measure in that assay. And you could look at the performance of the standard curve between each and every to make sure that they're comparable. You could basically overlay them in our software, look at things like recovery, the accuracy of your recovery, look at percent CVs to make sure that everything is basically looking very comparable between plates. There's also an assay control that's oftentimes included in kits where you could look to see that it's always falling within a range that you want. But in terms of ideal controls, I think that labs, it would be nice for them to develop a biologically relevant control. So potentially if they're studying a disease, a sample that is a disease sample and a non-disease sample to look at basically the different ranges of the analyte that they're interested in looking at. that is also most similar to the samples that they'll be testing. And you can add that to the assay control as sort of like a check in the range in which you feel like your samples are going to be falling. And then you can analyze that day to day and trend that to make sure that it's always falling within a specific range that you're comfortable with in terms of variability.
Yeah, I would add also really the controls that you use are either going to be your standard curves, assay included controls that are lyophilized daily that may be provided by the manufacturer, some kind of spiked protein control that you create, maybe in a relevant biological sample, or just a patient exhibiting what you're looking for to serve as a control. And in a general sense, you know, any of those might work, any of those could be good for your type of study. But as you run these assays day to day, in practice, you find very little variability over days. You know, your standard 2 that may have a median fluorescent intensity of 40 or 50 one day, it's going to have that same kind of level. It's not going to be 500 the next day. So you can overlay the standard curve for multiple days within the software, verified at that level. But then obviously, like with many tests or ELISAs and so forth, just sensible controls are pretty much what you would use.
Yeah, and actually, Rick, you bring up a good point. You had mentioned, you know, you want to reconstitute the control daily. So you don't want to use, and I think Brett brought this up in a prior question or an answer to a prior question, but you don't want to use a control that's been sitting in 4 degrees where the proteins may not be in the same state as when you first freshly reconstituted it. So if you're planning on, say, using you want to use one sort of control all throughout without having to reconstitute something every day. You might want to pool a bunch of standards. You could buy these standard vials, go ahead and dilute them to a certain concentration, freeze them at -80, and then pull one aliquot out each day if you don't have a biologic control that you've aliquoted out yourself. So you want to minimize the number of freeze thaws, which is really important in terms of making sure that you're looking at the same sample every day. And degradation not contributing to, degradation's not skewing your results.
Yeah, great points. All right, so I think this is a good next one then, talking about looking at samples across different days. So is throughput an issue? And what about data management? You kind of talked a little bit about it before, but.
Yeah, I think I can take this one. So yeah, the throughput is not really an issue because of as I explained earlier, the ability to scale the process. I guess if you're watching this, you probably love data or have a love-hate relationship with data. We know we're very familiar with that. Luminex does multiplexing does generate quite a bit of data. So with that in mind, we've obviously developed our software so that you can easily manage that data and quickly visualize and organize your data. So we have Bio-Plex Manager, which controls the instrument. It has automatic features for curve optimization. So there's settings that identify outliers for you. So you just click a button, you can analyze all of your standard curves at one time very quickly. You can also export it to Bio-Plex Data Pro, and that's a database background software that without any fear of modifying your original data, you're able to add columns, characteristics to the data set, and then you can combine different plates. You can overlay standard curves, and then visualize by groups and heat maps. And so there's just a lot of flexibility in that software and how to deal with just mass amounts of data quickly and easily. Yeah,
I imagine when you're doing a 48-plex assay, that's a lot of data to look at, and a lot. If you're doing 78 samples at a full plate, that's a lot. But hey, some of us like data, right? So you know, could be a good week or so, just your head down with your lots of coffee and tea. All right, here we go. One more question has come in. So for the panel, could you elaborate a little on the choice of clonality of antibodies for selection of capture and detection? What are the trade-offs of using a polyclonal for capture? Good question. Who wants to take this one? Candice.
Sorry. I mean, just, I can start, but feel free to chime in, guys. So in terms of clonality, oftentimes we'll try to use a monoclonal antibody for the capture just because you want to make sure that you're going to get reproducibility between different builds and between different lots. You know, as a vendor, we want to provide very comparable data for our kits. And so that's a major concern in terms of being able to provide that reproducibility. With polyclonals, it's not something that we would be against using, but the considerations there as a vendor for these type of assays would be that you might not get lot-to-lot reproducibility if that animal dies, you know, in terms of the same characteristics for the next polyclonal lot from a different animal. And so those are some considerations just in terms of reproducibility. But really, if the assay results look much better with a polyclonal antibody, I think that going with it is probably fine. If you had a choice between, say, if it's a sandwich immunoassay and you had the choice between using the polyclonal for detection or for capture, you'd want to probably use a monoclonal for the capture and then use the poly for detection. So those are just some considerations. For us, we want to provide the best, you know, assay format and best, you know, signal and result and working range. So we'll use the antibodies that are going to give us that response that we're looking for. But we also have to make considerations about reproducibility, which are less with polyclonals when you are looking at different lots.
Yeah. Yeah, I would add that the answer to that question might be a little different if you're talking about the corporate level in designing assays for commercial use versus a homebrew assay. If you've got a couple in hand, it's best to try them in different configurations for a homebrewed or custom assay to see which is going to give you the best sensitivity. And then, of course, you know, how consistent is that access to that particular antibody over time if you want a couple more assays and so forth. But yeah, it's really about the sensitivity. There's nothing wrong with polyclonals, but obviously if you're going to be mapping or looking for specific peptides of a given protein, you may want to obviously make your choices based on that.
Yeah, you might just want to do more studies in terms of between lots to make sure that the characteristics that are most important to you for the polyclonals, when you're using polyclonal antibodies, that it matches and meets your criteria. So yeah, I don't have anything against polyclonal antibodies or use of them.
Poor polyclonals. All right, here we go. I'm going to have to make this the last one, I'm afraid, because we are closing out on time. So for the big finale, everyone, has anyone used these assays in their vaccine development? So I guess, has anyone used Bio-Plex assays for vaccine development that you know of?
I can start with it.
Go ahead,
yes.
Candice.
You know, there are several publications out there of these assays or the raw beads with the amine coupling kit being used. So if it's a serology assay, they're going to be using their own antibodies and antigens, or I guess their own raw materials and coding it on the bead. But you're using the same platform. And then if they're looking at, you know, immune response profiling, then they can go ahead and use the off-the-shelf kits that exist out there for, you know, cytokines and other targets. But they've been used for both. So some of the examples that I could think of just, you know, off the top of my head, I think I'd mentioned Gardasil earlier. So the amine coupling that you do in the carboxy beads were actually, you know, a part of their development from preclinical all the way through clinical trials. And there were even some aftermarket studies that I read about. And so these are being done by some research institutions sometimes, but it can definitely be used throughout the whole development process. And then I think we had talked about looking at whether or not you're eliciting the immune response that you intend. So using cytokines to monitor as you're developing your vaccines, whether or not your host is eliciting the immune response you expect. You could use that from the preclinical to the clinical. I think we talked about the rat and mouse being models that can be used in the preclinical trials. And then for our human cytokine assays, you could use that during the clinical 1 through 3 phases. but yeah, definitely publications out there. Of course, we don't know every single person out there that's using it, because some institutions are quiet about their use of materials.
I was going to say, but Gardasil's a pretty well-known vaccine, so hopefully that helps. All right. Well, I guess looking at time, does anyone have anything to add before I close us out on that question? I didn't want to cut anyone off. Okay. No. All right. Well, okay, everyone breathe a big sigh of relief. You are done. Thank you so much to Brett and Rick and Candice. Great job. Hopefully now you can all go and have a lay down or a coffee or whatever you need to decompress after this grilling for an hour. Great conversation, some really good points there that hopefully is going to help a lot of the people in the audience with their own research. So before I do the official sign off, I just wanted to give a quick plug to some other webinars that are available in the Coffee Chat series. So we have recorded quite a few already. So if you would like to listen to those on demand, please go ahead. And if you want to sign up for some additional ones that are going to be live, then we have a droplet digital PCR in cell and gene therapy coming up, a real-time PCR in infectious disease, pretty topical right now with everything that's going on. A flow cytometry one, helping you develop multi-parameter flow panels, and then one on mixed mode chromatography. So a little bit of everything, hopefully to tickle your fancy, as they say. And with that, I will sign off and just thank you again to our great panelists, and thank you everyone for listening in. Take care and stay safe. Bye-bye.